when you look at an animal from afar, you say, well, it's no longer alive. And on some level, you're right, that animal is not alive. But on a different level, it's very much alive.
Have you ever wondered what happens to you when you die? In this documentary, we will tackle decomposition and nutrient cycling. So stay with us, we're about to share 8 ways to make things easier for you guys. Point 1. Cycling of nutrients represented by the nitrogen cycle. Nitrogen cycle describes how nitrogen moves between plants, animals, bacteria, atmosphere, and soil. Nitrogen to be used in different life forms on Earth must be changed into different states: ammonium, nitrites, and nitrates. 78% of air in the atmosphere is composed of nitrogen, but this is not usable in plants because it is unreactive. Thus, nitrogen is converted to nitrates to be usable. So nitrogen cycle goes like this.
Your heart is broken to your surprise. You're sick of crying for blue eyes. So tired of living misunderstood. Think you're a woman, I think you should. Come, sorrow is so peculiar. Comes in a day, then it'll never leave you. You take up it, wonder if it will fix you. Then wonder why sorrow has never left you. I'm talking about blue eyes, blue eyes. What's the matter, matter, blue eyes, blue eyes. What's the matter, matter, so blind, so blind. What's the matter, matter, blue eyes, blue eyes. What's the matter with you? Your heart got broken on the underground. Go find your spirit and lost and found. Oh, I've been watching how you behave. Not much like a lover, more like a slave. Come, sorrow is so peculiar. Comes in a day, then it'll never leave you. You take up it, wonder if it will fix you. Then wonder why sorrow has never left you. I'm talking about blue eyes, blue eyes. What's the matter, matter, blue eyes, blue eyes. What's the matter, matter, so blind, so blind. What's the matter, matter, blue eyes, blue eyes. What's the matter with blue eyes, blue eyes. What's the matter, matter, blue eyes, blue eyes. What's the matter, matter, so blind, so blind. What's the matter, matter, blue eyes, blue eyes. What's the matter with you?
Point 2. Different organisms aid in decomposition. To obtain ammonia from dead plants, there is a need for decomposition. This is aided by invertebrate detritivores, microfauna and macrofauna, mesofauna, and macro and megafauna. Point 3. Organic matters decompose at different rates. And this is because decomposition rate is influenced by several factors. One of these factors is the quality of plant litter as a substrate for microorganisms and soil fauna. Carbon is plentiful in plant remains, typically 45 to 60% of the total dry weight of plant tissues. Not all carbon compounds are of equal quality as an energy source of microbial decomposers. There are 3 known energy sources. One is glucose and other simple sugars, which are physically small molecules that possess high quality of carbon. Second, cellulose and and hemicellulose that are known to have more complex structures and therefore more different to decompose. And third, lignin, which are the most complex and variable carbon compounds which are broken down by only a single group of fungi, the Basidiomycetes, which include the mushrooms. Carbon quality of plant litters can have a particular importance influence on decomposition in coastal marine environments. There is an inverse relationship between the decomposition rate for plant litter and its lignin content at the start of decomposition. The other factors are the features of physical environments such as soil properties, which include texture and pH, climate, which include temperature and precipitation, that greatly influence microbial activity in such a way that microbial activity is inhibited at low temperatures and dry conditions. Optimum environment for microbes is warm and moist. Therefore, decomposition rates are highest in warm and wet climates. Alternate wetting, drying, and continuous dry spells tend to reduce microflora activity and populations.
Understanding plant litter decomposition is a key part of understanding the amount of carbon in the soil and its role in global warming. In order to appreciate the process of plant litter decomposition, it's important to understand the anatomy of a leaf. Leaves are made of many specialized cells, which determine its shape, size, and function. Surrounding each cell is a cell wall, and between each of the cells is an interstitial space. The cell wall surrounds the cytoplasm and intracellular components, most of which are leached out shortly after leaf fall. The cell wall supports the overall leaf structure, protects the cell, and separates the intracellular components from the extracellular environment. The inside layer is the plasma membrane. In between these two layers is the primary cell wall, which is the main target of decomposition. The three main components of the primary cell wall are cellulose, hemicellulose, and lignin. Cellulose consists of sugars known as glucose. Each individual sugar molecule is linked together to form a long linear polymer. These long linear polymers group together to form a crystalline structure. Since cellulose consists of sugars, it's a high-energy food that microbes attack first in decomposition. Hemicellulose is a polymer made up of chains of sugar molecules, primarily xylose and arabinose. These are also high-energy foods. Hemicellulose consists of shorter chains and has a branch structure. Hemicellulose encases cellulose to form a microfibril and plays an important role in holding everything together. Lignin is a complex compound made of different kinds of phenol-based molecules, which are arranged in random patterns to form large polymers. Lignin traverses the spaces between cellulose bundles. It's a low-energy food, which requires a lot of work to break down and shields the high-energy food of cellulose and hemicellulose. Microbes are microscopic organisms that belong to groups such as fungi and bacteria. They are the most numerous organisms on Earth and are sometimes referred to as germs. They can live everywhere, from air, soil, vegetation, and water to the human body. Microbes play an important role in almost everything, including leaf decomposition. During decomposition, microbes break through the leaf cuticle and traverse the interstitial space on their way to the cell wall. Microbes find food by releasing a small number of enzymes that break down complex compounds, like cellulose, into simpler ones, such as glucose, that can then be consumed. An increase in the concentration of these simple compounds tells the microbes to produce more of the needed enzymes for more release. The three main structural components of a leaf that microbes break down are cellulose, hemicellulose, and lignin. Microbes attack these high-energy foods, such as cellulose and hemicellulose, first. Lignin is a low-energy food, which shields cellulose and hemicellulose. Microbes only attack lignin as a last resort to get the protected high-energy food. There are two methods enzymes use to break down large, complex compounds into simpler, smaller ones that they can eat. One is the lock and key method, which is used to break down cellulose and hemicellulose molecules, which are chains of sugars, into single sugars that can be consumed. In this process, the enzyme is the key. The key fits the lock on the matching substrate molecule and causes a break in the chain. Some enzymes use this method to break a long chain in the middle to create shorter chains. Other enzymes clip off the ends to release individual sugars. Typically, multiple enzymes are packaged together into a complex scaffold. The scaffold organizes the enzymes that break bonds on both the ends and the center of the chain. To completely break down the chain, the scaffold slides up and down the chain and takes it apart. The other way enzymes break down complex compounds is by oxidation. This process is used to break down lignin. Lignin has a random structure. Every molecule is different, so the lock and key method does not work. Also, it takes a lot of energy to break down lignin, which is a very stable molecule. Because of this, enzymes do not have enough energy to attack lignin directly. Instead, enzymes attack other molecules that create free radicals, which are highly reactive compounds. These free radicals then react with the lignin and break it down. Once the lignin is removed in this way, the cellulose and hemicellulose that was encased by the lignin is available for decomposition. As microbes consume these foods, they are releasing carbon dioxide into the atmosphere through the process of respiration. Scientists need to understand how leaves decay and what influences how fast this process occurs so that they can predict how much carbon dioxide will be produced during decomposition.
Just like the quality of carbon compounds as an energy source for microbial decomposers, nutrient quality of dead organic material based on the nitrogen content varies greatly. Most dead leaf material has a nitrogen content in the range of 0.5 to 1.5%. The higher the nitrogen content of dead leaf, the higher the nutrient value for the microbes and fungi that feed upon the leaf. As the dead organic matter is being consumed, the microbial decomposers, the bacteria and the fungi, transform nitrogen and other elements contained in organic compounds into inorganic or minerals form. This process is called mineralization. Let's take ammonia as an example. Ammonia is the inorganic form of nitrogen and it is the waste product of microbial metabolism. The same decomposers that are responsible for mineralization also require nitrogen for their own growth and reproduction. Immobilization is the uptake and assimilation of mineral by microbial decomposers, and it occurs whenever mineralization occurs. Both of these processes take place as decomposer organisms consumes the litter. Net mineralization rate is the supply rate of mineral nutrients of the soil during the process of decomposition. Changes in the nitrogen content of plant litter during decomposition typically proceeds in 3 stages. After the initial period called leaching, nitrogen content typically increases as microbial decomposers immobilize nitrogen from outside the litter. Thus, because of this, nitrogen concentrations can rise above 100%, actually excluding the nitrogen content of the litter material. As decomposition proceeds and carbon quality declines, the mineralization rate exceeds the immobilization rate and the result is a net release of nitrogen to the soil as water. Point 5. What is the rhizosphere? The rhizosphere is a zone of soil surrounding a plant root where the biology and the chemistry of the soil are influenced by the root. It is an area of intense biological and chemical activity influenced by the compounds exuded by the root and by microorganisms feeding on the compounds. The roots exudes water and compounds broadly known as exudates. Here are 7 exudate functions. Number 1. defend the rhizosphere and root against pathogenic microorganisms. Root cells are under continual attack from microorganisms and survive by secreting defense proteins and other antimicrobial chemicals. Attract and repel particular microspecies and populations. High levels of moisture and nutrients in the rhizosphere attract much greater numbers of microorganisms than elsewhere in the soil. Number 3. Keep the soil around the roots moist. Research has found that rhizosphere soil is significantly wetter than bulk soil, which protects roots from drying out. Number 4. Obtain nutrients. The exudates help root absorb and store ions for plant use.
To be successful, iron must be available in a soluble form. It cannot be absorbed by the plant without first being made soluble.
Number 5. Change the chemical properties of the soil around the roots. The rhizosphere environments generally has a lower pH, lower oxygen, and higher carbon dioxide concentrations. However, exudates can make the soil in the rhizosphere more acid or alkaline, depending on the nutrients roots that are taking from the soil. Number 6. Stabilize soil aggregates around the roots. Sticky mucilage secreted from continuously growing root caps cells is believed to alter the surrounding soil. Number 7. Inhibit the growth of competing plant species. Plant roots are in continual communication with surrounding root systems and quickly recognize and prevent the presence of invading roots through chemical messengers. This process is known as allelopathy. Point 6! As decomposition occurs in the soil, decomposition also occurs in the aquatic environments. An important component of the aquatic nutrient cycle is the microbial loop. Here is a clip to explain this phenomenon.
The microbes in the ocean are the very small organisms that we can't see with the naked eye. We start with the smallest, they're the viruses, very, very tiny. And if we take a teaspoon of water as a measure, then we're looking at many millions of viruses in a teaspoon of water from the ocean. And if we go the next step up, we start to think about the bacteria. Again, very small, microscopic, around about 2 to 5 million in a teaspoon of water. If we go to the next size up, we're starting to look at the algae or the small single-cell plants that we call phytoplankton. And in a teaspoon of water, we're looking at several thousand to maybe 30, 40, 50,000. And then if we go the next step up of the microbes, we get into the very small microscopic animals that eat the bacteria and the small phytoplankton. And there we're looking at, in a teaspoon of water, 1 to 2 in a teaspoon of water. The microbial loop in the ocean is all the microbial organisms and their relationships to each other in terms of their interactions in the food web. The microbial loop forms the very bottom of the food web with all the microscopic organisms. So we have the bacteria taking up organic material from the water and growing. We have the algae using sunlight to photosynthesize and grow, and then we have the microzooplankton that then eat the bacteria and the algae. And those microzooplankton then release in their feces nutrients like nitrogen and phosphorus that are really important for the growth of the bacteria and the algae, and they recycle that back into the system. We've got to remember also that once we get out of that microbial loop, then those small microzooplankton are eaten by larger zooplankton, and that then goes up through the food web to the larger organisms, to fish and marine mammals in the Southern Ocean. Also, you get the cycling of carbon in the water, so you get release of organic carbon by the algae and also the microzooplankton, and that's taken up by the bacteria, so you get quite a quick cycling of both nutrients and carbon within the microbial loop. And that's really important in terms of how fast things are moving and cycling within the ocean. And also, when you have a dominance of the microbial loop in the ocean, you tend to get a lot of recycling in the upper waters. and then you don't get as much carbon, the big algae and the larger zooplankton growing with lots of carbon sinking out into the deep ocean, what we call carbon sequestration, where you take carbon out of the atmosphere into the water, it's used by the algae and then taken into the deep ocean.
Point 7. Nutrient cycling differs between terrestrial and open water aquatic ecosystems. Nutrient cycling is an important process that represents a direct link between net primary productivity and decomposition. In terrestrial ecosystems, plants physically exist on both the decomposition zone at the soil surface and the productivity zone in the plant canopy. In an open water aquatic ecosystem, the zones of decomposition and the benthic zone are physically separated from the surface waters, where temperatures and light availability support primary productivity. Before we head to the last point, let's have a recap. Point 1. Cycling of nutrients is represented by the nitrogen cycle. Point 2. Different organisms aid in decomposition. Point 3. Organic matters decompose at different rates. Point 4. Quality of nitrogen. Point 5. The rhizosphere. Point 6. As decomposition occurs in the soil, decomposition also occurs in aquatic environments. Point 7. Nutrient cycling differs between terrestrial and open water aquatic systems. And finally, point 8. Now down to our last point. Yes! Nutrient spiraling in streams and rivers. Jack Webster at the University of Georgia noted that nutrients are being transported downstream in a spiral rather than a cycle. The process of recycling, retention, and downstream transport may be pictured as a spiral lying horizontally in a stream. Uptake and turnover occur as nutrients flow downstream. The longer the nutrients remain in place, the tighter the spiraling, and the shorter the distance, the tighter the spiral.
This is the story of a watershed, which, it turns out, is about more than water. That's because a watershed also cycles nutrients up and down and in and out of headwaters, streams, and rivers that supply clean water for plants and animals. How does that happen? Well, let's take a closer look. It has taken the water centuries to carve this path, flowing over rocks, washing away the soil, and even knocking down trees. All that tumbling water adds oxygen to the stream. The shade keeps the water cool. And all of that makes it perfect for a raccoon, which on this early October day is getting a drink. A water thrush joins the raccoon. It snaps up a fat stonefly. It's going to need that energy for its long migration to Central America. It is a sunny day, but an early cold front has started the leaves falling. Let's see what happens. In a few days, a dark film of bacteria covers the soggy leaves, and Onchotarsus comes to examine this delicious discovery. It is a year-old larval toe-winged beetle. It does not know that predators, like the stonefly Ecoptura, prowl leaf packs looking for herbivores. For the Ecoptura, it's a successful hunt, but even it doesn't see the school of rosy-sided dace, a kind of minnow, swimming nearby. These fish travel the length of the river basin, from the headwaters to the streams and the rivers, and back again in search of food and shelter. In the food chain that supports the complex web of life throughout the watershed, what is lunch today becomes waste tomorrow. But the waste is full of nutrients like nitrogen and phosphorus, calcium and potassium. For example, the kingfisher's guano lands in the water and disperses a cloud of nutrients. Tiny microbes in the water attach to the guano and begin multiplying. The tiny food particles are a perfect snack for a caddisfly named Hydropsyche. It spins a web to catch food particles. But living nearby is another type of caddisfly. Rhynchophila is a predator, and it has been waiting. It begins to attack, but all the commotion catches the eye of a brook trout, just in time for Hydropsyche to escape. The brook trout plays a role in the nutrient cycle, and not only as a top predator. You see, as the fish swims away, the mucus-like slime that protects its scales sloughs off into the water. It is full of nutrients, and the microbes in the water quickly move in. As the microbe colony settles to the bottom of the stream, the brown mayfly Macaphritium settles in for lunch. It also leaves behind what is left of its breakfast. Now, let's go back to the surface of the stream for a minute. You see, where the water flow is really strong, in front of a boulder and the curve of a stream bend, some of the water gets pushed through the porous bottom of the stream bank where it mixes with the groundwater beneath the channel. That's how the nutrients in that bit of breakfast the brown mayfly left behind end up downstream. The mixture travels for a ways underneath the channel of the stream and emerges into a large sunlit expanse of river water. The nutrients, warmed by the sun, fuel an explosion of single-celled plant life in the river. Silica-shelled diatoms feast on the nutrients. They use the nitrogen, the phosphorus, the calcium, and the potassium to maintain the chlorophyll, the amino acids, and the nucleotides that drive their molecular machinery. The diatoms are a delightful discovery for a hungry crustacean called Daphnia, which uses its feathery antenna to filter out the tiny diatoms. At night, the satisfied Daphnia drop to the bottom, only to provide dinner for an Eshna, which it turns out is very hungry because it is getting ready to molt. It climbs out of the water and slowly sheds its exoskeleton to be transformed into an adult mosaic darner dragonfly. It will use those new wings to fly up to the headwaters where our story began. Along the way, it will find a mate. When it lays its eggs, it will have recycled nutrients from the bottom of the watershed to the top. Clean water, you see, provides and protects all this biodiversity, and the biodiversity helps ensure clean water. It's an intricate, life-giving choreography that can only be staged in a healthy watershed.